The final speaker is Keith Whitfield, who is president and professor of psychology at the University of Nevada at Las Vegas. His talk title is Advantages of Within-Group Analysis of Race Ethnicity.
Well, good afternoon, everyone. Can you see that now?
Yes.
All right. Well, Frank, I thought you were going to introduce me as a former Dookie.
Okay, I didn't do that.
Yes, a number of years ago. Seems like it's a number of years ago. I was there at Duke, sitting in the awe of some fantastic people who helped me with thinking about what I'm going to share with you today. This is actually over a lifetime of a career of thinking about how we actually study, understand race and ethnicity as it applies to lots of things relative to aging. And I think that it's quite pertinent as we think about it relative to ADRD. So sharing with you this citation, and it's a little long in the tooth, but I'm actually working on trying to do an update of it. But it asks the question, are comparisons the answers to understanding behavioral aspects of aging in racial and ethnic groups? And, you know, that came from a lot of thinking about most of the time when I would see research, it was always comparative research. And the research that I was doing was focused specifically on African Americans, and got lots of questions about it and, you know, applicability. We're going to talk about all of those things. The previous presenter said that she didn't have too many data slides. I think I have even fewer, but I think I really want you all to think about this as a concept, as a way of thinking about how we understand race ethnicity as it applies to ADRD. So, you know, our little graphic here kind of thinks about aging and making sure that we look at it through a cultural lens, that some of the things that we look at relative to sensory systems or physical health, mental health, just some general areas, it may look the same as you compare one group to another, or it may be slightly different, or it may be that we see one pattern and see a very different pattern in another group. And so we always have to make sure that we keep that cultural lens on. But once we do that, what does that mean in terms of how we approach, how we think about, analyze, or make interpretations even about our data? So I just provide this for you, you know, total digit symbol score and what we find as we age. Of course, that we find that there are reductions in terms of performance as you go across the decades. But it's interesting to note, you know, if your statistic professor was there, you know, the first thing they tell you to do is that you should plot your data. And if we plot our data for that exact same thing, we find incredible variability. Now, I know I'm preaching to the choir. I know you guys get this, but this is something that it's at the heart of as we think about between group comparisons, of just remembering that there's incredible variability when we look at any group. And this actually happens to be some data from African Americans. Now, here is something that came from Americans Changing Lives. And if you look on the right-hand side, you find some data on their measure of cognition. It's a funky kind of measure. But then when we sit and plot the data and look at African Americans, you can see it, that there is greater variability in African Americans on that particular measure. And even if we do a test of homogeneity of variance, what do you find? That the two groups are not homogeneous in their variability. And actually, your basic statistics professor will tell you that you're not even supposed to compare those two, that you're violating one of the assumptions of variance that underlies performance. So if we take this and then put it in the context of thinking about studying dementia, studying cognitive functioning, I think that this is a very simplistic way of thinking about it, of that, and actually across the course of my career, I've really appreciated understanding and seeing MCI come into the factor, because I would find people when I was studying normal cognitive aging that, you know, that decision point, of that clinical threshold wasn't a clean clinical threshold. And MCI in some ways shows that yes, there is this trek or course of trajectory that happens from normal cognitive aging into dementia, and that we need to pay attention to it. But as we take this and then think about it in two different groups, there's interesting possibilities that we should consider. For example, it may be that that course progression of MCI as it relates just to age in the simplistic presentation may actually end up looking the same across groups. But it also may be different, and we can talk about some of the reasons of why it may be different. Maybe it's shorter. Maybe you have a set of normal course of normal cognitive aging, and then you hit those things with deficits in physical functioning and other things that go into MCI that then lead us much more quickly into a state of dementia. Or it could be that it's a little bit longer, that what we consider normal cognitive aging actually is a shorter course, and then that there is a longer course of MCI due in part to chronic conditions and other things that occur in higher prevalence in African Americans that make the difference between the patterns that we would see across two groups different from one another. The variability is going to show us some of that. And so what I'd like to do in this presentation today, and I'm going to see if we can have plenty of time for discussion because this typically, solicits a lot of discussion, is just I'm going to go very quickly and just talk about kind of the scientific process of that as we're thinking about this, as you're applying it to whatever you may be studying relative to ADRD. There's things where you should consider how culture may impact at each one of those steps. So let's talk about questions first. And so the first question is what is the question? And typically we're asking, how do we typically understand minority populations? We usually ask two different kinds of questions. Are there differences in cognitive performance by race? Or what factors account for differences in cognitive performance? But what's really garnered from comparisons research when you're asking the question in that way is that that there is or is not a group difference. And that we even try to get at that after looking at covariates that are controlling for what the difference may be. But remember, what we're getting from that is that there is a difference or there's not a difference. We're not explaining anything too deeply about what drives the difference. Why is there a difference? I think needs to be more of our question that we pose. So if we're really interested in the minority group, there needs to be a different kind of question. You know, what factors account for understanding for individual variability in African Americans, let's say, or for between group analyses to be more meaningful? What makes African Americans and Caucasians different in cognitive functioning, and the why? It's the that we oftentimes do not talk about or put into our explanatory models. And I'll give you a couple of examples how you can do that, I think, a little bit later here. But that's the big point. Performing analyses that provide explanations for differences in variance and not just the difference in variance. Those are different and much more elucidative of and illustrative of what may really be going on as we try to understand racial or ethnic differences. We can also think about how culture impacts the model or approach that we're taking. There's a wonderful paper, that paper that I showed you earlier, and I can go back to it if you want to find the citation. But it drew from a paper by Kause, Coronado, and Watson that was taking a look at different models. Models being how we're thinking about how cognitive, or well, in this case, how cognitive differences actually arise or how we explain them. And so one is using a cultural deviance model. And I think that this is very old thinking, where we see that differences or deviations between groups are seen as deviant or inferior. One would think that that is a model that is far out of understanding, but I occasionally see papers that have that written into the interpretation of the results. Then there's what I like to consider the 70s model of cultural differences, and that's looking at a cultural equivalence model. Using this sort of model, lots is placed on superior economic status, has provided advantages, and then produces differences as you look at two different groups. The deviant perspective, on the other hand, contributes them to the actual culture of the group. And then there's the cultural variant model, which is, I think, the more appropriate, more nouveau model to be using to think about differences. And that in this model, explanations end up being discussed as resilience in the face of oppression, adaptation and survival due to external forces, or that the differences are culturally rooted internal explanations for differences. That people have come to where they are, even the variability is driven and influenced by factors that may be culturally rooted in what that group is or what they experience or what they have as, in many other cases, are thought of as strengths. So now let's talk about the measurement and a little bit about recruitment. So one of the things that is challenging is trying to have the same sampling approach across groups. Now, I know our previous speaker actually talked about Enhance, which I think did a really good job of recruiting people using a similar approach, but actually really oversampled so that they would get a representative sample of minorities. This is something that ends up being very hard to do. There's certain strategies, but they have to be ones that are thought of to be useful. And one very interesting example of the difficulty was what I know of from the work that was done in the Detroit Active Study, where they started off thinking, well, we'll recruit African Americans first, and we'll get them because they'll be the hardest ones to do. But the structures and the approaches that they put into that recruitment did not easily translate into what they could do in terms of recruiting whites in the city. And so they actually had to change their approach ever so slightly. So that's a great example. Now, is there equivalence in measurement across groups? And are there correction factors applicable? You know, this is something that I feel like I'm aging myself these days because I keep thinking about the old days, and that you used to see lots of examples of papers that would come out that would demonstrate the measurement of different evaluations. Like for the MMSE, we would see how they work for different groups, and people do latent class analysis. They do lots of things to really get at the heart of whether those measures look like they're measuring the same thing across groups, but you don't see very much of that anymore. But there's lots of different ways that we can think about equivalence. We can look at cultural equivalence, conceptual equivalence, linguistic equivalence. Linguistic is probably the easiest one there. The idea that the language of items is understood to be the same across groups. I have in here an example of blue, and it's from back in the old days when I first started working. I was using the CESD, and I was down in Louisiana, and I gave the CESD, and on the item that asked, "Are you blue?" almost 80% of the folks would give the highest number, said yes, yes, I'm blue. And I thought, oh my gosh, there's lots of folks here who have who are depressed. If we use that as a single item, it would show that people were just massive numbers or massive proportions were depressed. But I wondered about it, and then I went and asked folks, and linguistically, for the folks that I talked to, when you ask about being blue, blue was a good thing, because it meant that you were engaged in some inner turmoil that ultimately was going to lead to you being better. And so blue was kind of culturally accepted of that. Of course you would have the blues. Just a corny example of how we can study different groups and that they may actually respond to what we're asking them or what we're asking them to do in different ways. Functional equivalence, scalar equivalence, other ways in which we can measure or we can assess how constructs are behaving, measuring the concept that we are interested across groups. So then we can talk about analyses. So first, let's talk about measurement error. I think all of you all know that precision is a measure of how closely the parameter estimate is expected to be a true value of the parameter. And just keep in mind that precision is driven in part by the smaller of the groups. And so we must use instruments that measure the construct of interest as equally as we can across those groups, or it's going to be weighted more closely to the smaller of the two groups. So just keep that in mind. I'm going to come back to that in just a second. So between group comparisons, typically the straightforward basic thing we think about mean differences, it's a reasonable elementary step to be able to understand what's going on in terms of our analyses. But variance differences are probably a little bit more accurate, and they're behind what's behind mean differences anyway. And that we can do very, creative sorts of things using structural equation modeling to assess invariance between groups that probably get us at much better ways of being able to assess the differences that we find between groups. We, of course, may need to covary for other factors to be able to fully understand, but that's very important. Also, in terms of analysis, I want to talk just a second about sample size and the power to detect differences. We need to make sure that the samples need to be just more than representative in size from which they're drawn from. The variances need to be equivalent to one another. And, the challenge typically comes from trying to find sufficient numbers of ethnic minorities to participate. Like we talked about in NHATS, they really did some super oversampling of minorities to be able to get reasonable numbers. And when we get down to some of our basic premises about analyses and talk about power, for power considerations, the larger the difference in the size of the groups, the larger the sample needs to be. So let me share with you a couple of examples here. So here's one, and as we look and see and look at, just this piece of a power curve, we come out to somewhere like 235 people. This is when we have one African-American for every 2 Caucasians, meaning if you had 50 African-Americans and you had a sample of 100, this starts to be what you have to get to be able to have the appropriate statistical power. Now look at what happens when we make that differential between the number of African-Americans and Caucasians larger. What happens is that our sample needs to continue to become larger and larger. That's when we're not being able to try to have equivalent size samples, that it's being driven that we have to have a larger sample overall to be able to get the appropriate statistical power. And so last in this example I want to share with you is about interpretations. And this is really how we think, and I think I'm sure we teach our students to make sure that they think that there's not always just one simple, single, straightforward answer for things. Differences can actually mean strength. And one of the things that I think the National Institute on Aging is doing a great job of doing is helping us make sure that we get multiple points of time, because trajectories of change may also be different, not just variance differences, but variance differences over time. So the more assessment points, the better. And I would even suggest that if you're really looking to try to figure out explanatory variables for trying to understand differences between groups, one might include a measure of ethnicity, something like acculturation, for example. It can be used to examine mediating or moderating effects that might be going on between groups, but something just to consider. Started off with this talking about between in contrast to within group. Just keep in mind that there's some great strengths with doing within group analyses where you're really just focusing on Hispanics or African Americans or whatever the group may be. And that if we understand, what we get from that is understanding the variability on measures specific to a group, and the error of measurement is limited to the measure, not to ethnicity, like it might be in between groups where the measure is not equally does not equally show up for both groups. The weaknesses that we have is that there's a limited description of the phenomena can be made, and description is specific only to the minority group. So I want to conclude with just a couple of points. One, should between group research be done? Some of what I just presented may suggest that it shouldn't be done. I think, of course, it should be done. Now, if I could take history and rewrite it, I might ask that we find as much within group research be done as between group research. I think that that is almost the opposite. That's now that we're understanding that as we've made comparisons, that far too often we really don't understand what the real difference between them is. We know that there is a difference, but why is there a difference? Also, which should come first, between or within? I'm a guy that's done mostly within group studies, and so I think they're important. And I always hope that the research that I've done actually gets used in between group research to be able to make sure that you're comparing apples to apples, that the kinds of things that influence one group, the variability that you're seeing in one group looks like it's the same kind of influences that you see in another. I do want to put in a pitch for studying minorities offers unique opportunities not yet fully appreciated by many researchers, both basic and applied, actually could be able to understand their phenomena because it's influenced by social or psychological factors. Future research should accurately and appropriately utilize minorities for understanding cognitive functioning and dysfunctioning, because I think that it will advance the field as a whole. Alternative strategies to studying minorities, both between and within. And, can you imagine, actually, I think David Bennett was on here earlier, and some of his group has done that, where they've done both between and within group analyses on some of the same data and found, interesting comparisons and interesting things to take a look at. I think that that's a great way to try to advance our understanding of not only minority people, but all people. And the takeaway is that there's more than one way to skin a cat, and that we should think about these other ways that we can conduct analyses and understand more about ADRD and the different kinds of factors, both social, psychological, biological, that create variability, and for us to be able to better understand the disease process itself. So with that, I will stop and see if there are any questions.
Keith,
Yes, sir.
this is David. How are you?
Oh, just fine. How are you?
I was hoping I would see you in the flesh here.
I tried, but they keep my leg pretty much so anchored to my desk here.
Oh, okay. Thank you very much. It's always enlightening to listen to you. I wonder if you can give us your thoughts about kind of best practices when you do have both within group and between group data. As you mentioned, we do a little bit of both. And, the within group is great because you can use all the data and you don't have to worry about all the pitfalls of doing the between group,
Yeah.
which there are quite a few. But and from a health equity point of view, I think that's fine. It doesn't, we don't always have to be focused on whether there's differences, but I always feel like I'm also missing something by not being able to address differences. And you can't when you just compare within group. You can see whether you have similar associations, but that's about as far as you can take it. So I wonder if you have any thoughts about best practices in this area and how best to approach it.
Yeah, and thank you, David, for teeing that up. And I do want to apologize. I noticed that I had it on speaker view, but I didn't know you all were seeing it on speaker view too. I apologize for that. But David, your point is very important, and it's not just cowing to the great one, but you're doing what a lot of people don't do, which is that from a health equity lens, they don't ever seem to look at the within group. All you find is race as a covariate that's there, it's significant, and there's no way to explain it. And if we don't start actually understanding what's behind it, all we're doing is perpetuating just that it's almost like that there's a gold standard and that everything else is just a deviant from it. And somehow that deviant is not important. That deviance, that difference that you find between it may be where some of our keys to actually understanding the course and trajectory, and even things that are factors that are most important in understanding disease processes are there. So to me, both of them need to be there. I don't think that while neither one is a single panacea in and of itself, you actually need both. So yeah, it's limited when you only use one group. Yeah, it's limited if you don't have something like a mediator, a moderator, or some way to be able to actually say that I have the explanatory variable that explains the differences between the groups. We're not at the point of understanding that for a lot of things. But variables like education, variables like, cardiovascular risk, there's lots of things that could be, but we tend not to do them. So I think that at this point in time, it's actually good using the approach that you're using, which is to be able to do both. It should just be that we're informed by both of them. Of that, so if you find a pattern of in African Americans, it's education and social support that are making for bad outcomes in terms of follow-up for seeing your physician, and in whites, it's education and income. You've got some differences there to be able to think about, and that they shouldn't just be jammed together. That what's going to happen is that if you've got, as I was showing with some of the power analysis things, what you're going to find is a set of results that's driven by the larger sample, but it doesn't completely explain what's going on. Does that make sense, David?
Yes, yes.
What can we do about other groups? I understand the African American Caucasian comparisons, but say we look at Hispanics, or we look at Asian groups, or and so the question is the group even meaningful? Like for Asians, you combine Japanese and Indian and Cambodians and Chinese, and and they have different cultures, and maybe different biological, genetic makeups and so forth. So
Yeah.
does it make more sense for African American and Caucasian comparisons?
Well, there are subgroups within African Americans as well.
I'm sure.
I was
Yeah.
just thinking about the study I have called the Carolina African Americans with State of Aging. And there's a little group in there. They're just not very big, so we don't worry too much about them. But they're Native American and African American. So,
Yeah.
you do find those subgroups. And so to your example, Frank, that's exactly a very interesting question to be asking. That, for example, if we're looking at Hispanics, do folks from Puerto Rico have a different set of ways in which they interact with the medical field, which I will say that they will, or even perhaps some, and so let's talk about, an elephant that ends up in the room and some genetics. And so remember, we're looking at population, we're talking about population level genetics and the frequency of certain genes or certain gene combinations. And so that may be different than it is in Mexican Americans or folks from Mexico. That's important to know. That may be us understanding where some of those things that are genetic influences, what happens with them in real life in real time. Of that, it's not just those genetic differences within a subpopulation, it's then how they come to this country, foods that they eat, ways in which they interact with the medical system. That's pulling apart all of the things that, there's a reason why we haven't found simple answers to understanding Alzheimer's disease.
Yeah.
And it's because they're embedded with all of these other environmental factors, as well as with genetic combinations.
Mhm.
And so to me, it actually makes interesting sense to see if we can see if some of those populations make more sense to do between group studies. But before we get there, we've got to understand, well, for me anyway, you got to understand them within to see what those relationships are, and then say, okay, let's compare them against some of these other groups that we know, and to see whether it is some combination of either population prevalence of genes, or is it some kind of cultural factor that's going on. For some groups, we've studied them enough that we know lots of things about their culture. Let's use that to our advantage to be able to understand and discern what's going on. It will give us more robust understanding of how those factors, and not just in one group, across all of them. I think this is the challenge that David was talking about. Will actually elucidate what those bigger challenges are across all the groups, if we understand that they're important in one, but they don't show up in another group. Yeah. And so then we can make effective comparisons, but to some degree, we have to understand them within those groups first, is at least my thinking.
Thank you. Yeah.
I have a question again. It's Florence Johnson, and I'm a second-year student at the University of Michigan School of Nursing. And my area of research is family caregivers of people who live with dementia in the community. I'm really interested too in this area of within race study. But I would like to know if you can expound upon that culturation. I don't know that I've heard that before, and it sounds very intriguing.
Yeah, boy, you're going to make me feel old of that. That used to be something that everybody studied, and particularly anthropologists would study culturation measures to see how, now I want to get it right, it's how much a new culture may have, or a culture may have taken on the characteristics of a host culture. So if it's somebody coming from Peru who came to the United States after one generation or two generation, how much do they actually take on some of the beliefs, style, whatever of that host culture? You can also look at a culturative stress, where it's that taking on or dealing with another culture actually provides stress for somebody. So there's some interesting ways to take a look at that. There's a little bit of that in my paper, and I'm always trying to get my citation index up. So if you take a look at Whitfield, 2008, that's in the Journal of Gerontology, you'll see some of that discussed, and that may help you think a little bit more about it. But I do want to take this opportunity. That paper was in part developed from talking to students who weren't asking the right question, and they wanted us to do a within group, but their committee said that they had to do between group. And I think in many cases it's because they weren't asking the right question. Because if you say, well, I want to know what the difference is between these two, well, that begs that you're going to do a between group analysis. But if you say, I want to look at the variability that drives differences or changes within one group, that's more of a within group. So the way that you ask the question, the way to pose the question is very, very important in terms of the analyses that you would pursue. Is there enough time for me to try to answer one more question, Frank?
Yes, just one more, yes.
Yeah, I saw that Timothy Hughes wrote, can you expand on the benefits of within group research versus planned stratified analyses in diverse samples? I think that those planned stratified analyses are fine as long as you understand where you need to be to get the appropriate power to detect differences. I think what I used to typically see, and this is an over exaggeration, but I'd see samples that would study, they would study 1,000 people, 900 would be whites and 100 would be African Americans.
Yeah.
I always have so much issue with that. It's like, so you only had 100, so that's 10%. It's not even relative to the population, which people make that an argument for. But who were those people that you got? Was that a selective group that decided to select themselves into your sample versus for the majority one, 900 people decided that they'd come out and do it. What was driving the difference that you found between those two? But also, again, because you probably don't have the ability to statistically look for reliable differences because of the sample sizes, it's something just to be concerned about. And so as long as those stratified analyses actually, or maybe it's not the stratified analyses, it's stratified recruitment, actually takes that into account, I think that they're fine to be able to look at. I just encourage you, take a look within group and just see what's going on within the group and see if the pattern follows the same as it does when you put them together or when you compare them to the other group.
Well, thank you very much.